Good afternoon, everyone. I'm Christy Carter. I'm a program officer from the National Institute on Aging in the Division of Aging Biology. And it is my pleasure to introduce today's speaker to the Wednesday afternoon lecture series. Our honored guest is Dr. Laura Niedernhofer, who is a professor of biochemistry, molecular biology, and biophysics, and the director of the Institute on the Biology of Aging and Metabolism at the University of Minnesota. Dr. Niedernhofer began her scientific education at Duke University, where she received her Bachelor's of Science in Chemistry. She then went on to receive a Master's in Physiology from Georgetown University and an MD and a Ph.D. from Vanderbilt University. She was also a postdoc fellow at Erasmus University in the lab of Dr. Jan Hoijmakers. Dr. Niedernhofer's research focuses on the fundamental mechanisms of aging, and her work has helped us to better understand how damage to the DNA can play a causal role in the aging process and bring about the onset of age-related decline and dysfunction. In addition, Dr. Niedernhofer and colleagues have demonstrated that aging in the immune system can, in particular, drive cellular senescence. And drive age-related phenotypes, as well as aging in solid organs. Dr. Niedernhofer is currently the PI or co-PI on several NIA-funded projects, including the Minnesota Tissue Mapping Center for Senescent Cells, a component of the Senescence Network Common Fund Initiative, a T32 training grant supporting both pre- and postdoctoral fellow researchers in functional multi-omics of aging research, and a leader of a PO1 project that examines senescent cells that drive immune dysfunction with age. I'd like to close by saying that she is an outstanding mentor of early career scientists at every level. She is the recipient of the Scripps Florida Society of Research Fellows Mentorship Award and has mentored dozens of scientists who have followed in very diverse and unique career paths, including clinicians, academic and industry researchers, teachers, science writers, and government employees. And so we are very, very pleased to have her here with us today to share her insights on the complexity of the biology of aging and how we can move the field forward towards effective geroscience approaches. And just a few housekeeping notes. Please see that the CMA code for today is 50113. And for those of you who are watching online on the videocast, please send any questions by clicking the Send Live Feedback button on your screen, and we will relay them to the speaker at the end of the lecture. And for those of you who are here, we have a small reception following just outside of the auditorium. So without further ado, welcome to Dr. Niedernhofer.
Well, thank you very much, Dr. Carter, for hosting me today and having me here. I am absolutely thrilled and honored to be able to participate in this lectureship. So as Christy said, I come from the University of Minnesota, and we've got a brand new Institute on the Biology of Aging and Metabolism, which we created and I co-direct right now. This has been supported by the state of Minnesota, and we work closely with the Mayo Clinic in all aspects of geroscience research. So I want to talk to you today about disease-based research or geroscience. I made the assumption that this is a pan-NIH group, so it may not be incredibly sophisticated for those of you who know senescent cells and geroscience already, but I hope you'll enjoy it. So the objectives today are for me to provide a definition, rationale, historic perspective of geroscience, and where I think the current status of the field is, define cellular senescence and hallmark of aging, and how we think it is a great target for clinical trials focused on geroscience approaches, but there's still a lot we know and don't know. And then illustrate the opportunities and challenges in geroscience and how we need to really foster multidisciplinary approaches, I believe, to tackle this. So my disclosures. I serve on a couple of scientific advisory boards, but I have absolutely no stock or financial interest, and I have had sponsored research agreements with Merck and Pfizer to explore geroscience. So I'm hopeful that Big Pharma will get involved. I understand that this is the Margaret Pittman Lectureship, and I have just been fascinated reading about her history. I mean, what a lot of firsts in a woman. Ph.D. in the 1920s from the University of Chicago. I mean, on a question, does Haemophilus influenza cause influenza? That's just amazing, right? Imagine what we could have used her help during COVID-19. During the war, she did a lot of work on optimizing blood transfusions, and her most remarkable accomplishment is becoming the first woman laboratory lead at NIH, where she served for a decade and a half. And then she still, late in life, went on to the WHO and has helped with multiple different vaccine developments and standards. So this caused me to go back and reflect on my training history. And these are the women I have had the great good fortune of mentoring during my career to date. You see many of them have gone on to be professors in academia. I'm really proud of two of them who are at NIH, illustrated in red. Several have gone on to industry. Many are doing clinical plus being professors, and several are still on their way to their own independent academic career. What I'm proud to say is not one of them has left science, so hopefully we'll have a lot of Margaret Pittmans ahead of us. So the weird title of my talk was A Tale of Two Cities, the book by Charles Dickens. But I think it says a lot about what's going on right now. I think this is truly the best of times in geroscience, because there is so much technology out there that we're really going to be able to tackle some big questions. It's the worst of times because of politics and all sorts of things going on, and a little bit of an anti-scientist sentiment out there. Age of wisdom, I think the trainees that I see coming up are going to be fabulous to take over this field, and we've got AI and all sorts of other opportunities. There's a lot of foolishness going on, and I'm not going to even describe what's local. But it's a period also of epic belief. I do think we can be very transformative in improving the health of the elderly, but we have to fight all the incredulity out there about science. So why is geroscience so important right now? So what this graph is illustrating to you is the number of individuals in the blue bars is 65 to 84 years old, and then the yellow is 85 plus. So we are in 2024, past the halfway mark of 4 decades, when the number of individuals 65 to 84 is more than doubling, and the number of individuals over 85 is quadrupling. So this is a real shift in our human population demographics, a lot more older individuals. This is also the first time in human history when the number of individuals who are over 65 are going to exceed the number of youth. And so this is really challenging, and it's going to change every aspect of our society if we don't really tackle this head on. Minnesota has already reached this stage, and I can tell you from my own personal experience, my mother's in a nursing home, and she requires 8 caregivers to take care of her. So how are we possibly going to manage all of this when we have so many elderly individuals and that population is expanding? So why is this happening? Well, it's not all grim at all. It's happening because of all the incredible biomedical research that's happened over the last century. So what this graph is showing you in the black bar is the age at death in 1900. And you see a lot of infant mortality, middle-aged mortality, and not a lot of people reaching in centenarian age. But by 2016, this has completely shifted. And this is because of incredible advances in public health, but also infectious disease research and treatment for chronic diseases like cardiovascular disease and diabetes. So now we can happily expect to live into our 80s, 90s, and beyond, but this is what's in what's called the red zone, which is a period of increasing risk of morbidity as we age out. So what is this red zone? It's really all of these chronic diseases that are very familiar and highly, highly linked to older age. And so that you can see in this graph that right around the 6th decade of life, the risk or incidence of these diseases goes up exponentially. So this is a huge disease burden as our population ages out. This is, excuse me, another way to look at it that is a little bit frightening, and that is most of elderly individuals are going to have multiple diseases of old age and not just one. So what the graph is showing as you go from, excuse me, light blue to dark blue, increasing chronic diseases. So I use the, excuse me, that's what happens when I talk too fast. Sorry. The vertical bar here is showing 65 to 69-year-olds. And what you see is half of them are going to have 2 or more chronic diseases, while 25% are going to have 3 or more. So this raises the question of, even if we cured a single age-related disease, would we really impact the health of our older population? And then you can even add here, by the time you get out to that group of 85-year-olds, which is quadrupling, the vast majority of them are going to have multiple chronic diseases. So I actually want to make the point, too, that this is a pan-NIH problem because disease survivors are also aging very rapidly. So I happen to study a number of, well, cancer survivors are one group, these pediatric disorders of genome instability. These kids used to live until 12, maybe 20, and now they're living into their 60s and 70s, but they look like they're 90. We see accelerated aging in diabetics as well as obese individuals, and even organ transplantation. You can imagine all of the chemicals that these individuals have to be hit with that cause accelerated aging. So I actually feel that we are underestimating the challenges we're facing with aging biology, and we really need to think about the entire spectrum of diseases and age ranges. So this is another slide that really caught my attention, and this is work by Jay Olshansky. And what it's showing here as just one example is the risk factors for cancer. So we all know that if you smoke or too much alcohol, bad diet, some infections will increase your risk of cancer by 10 to, say, 35%. But if you were to put this on a scale of simply being 65 or older, you see that old age dwarfs everything else by orders of magnitude. And the same is true if I graph this for Alzheimer's disease or for heart disease. So what this means is we are developing drugs to treat all sorts of things that are actually minor risk factors for some of these chronic diseases. And it raises the question, could we find something here to target fundamental biology of aging that would be able to then attack all of these diseases in suite? So this is the geroscience hypothesis. We would like to develop therapeutics interventions that target fundamental aging biology rather than treat individual diseases of old age. And the impact here is supposed to be really dramatic. So the thought would be that with a treatment for aging biology, you could prevent, delay, or ameliorate multiple of these chronic diseases of old age that are degenerative and rob people of life quality and independence. This would help you avoid spending the rest of your life with all sorts of specialists, your cardiologist, neurologist, you name it. This would also then help you avoid polypharmacy, because oftentimes these specialists are not talking to one another. And so you get drug-drug interactions if the doctors aren't talking. And the side effects of this are actually a huge risk to older persons. And finally, this will have a huge impact on healthcare costs, as I'm sure you can imagine. So this is what we're after. I do want to recognize the leadership at NIH that has really tried to tout geroscience and bring all of NIH together as part of a geroscience interest group led by Richard Hodes, Felipe Sierra, Ron Kohansky, and Viviana Perez, Ned Sharpless, Dina Singer, Kevin Howcroft, and Mercy Prabhudas. So I'm sure I'm missing others, and I apologize if I have left you off. But these have been my role models in really focusing on this aspect of science. So what is the goal of geroscience? And this is a really, really important point. And that is, this is a graph that shows as you age out along the x-axis, you remain fit for a period of time. But there's usually this unfortunate slow slope of declining fitness. So with geroscience, what we want to do is simply square the curve. So what that means is, as you go out, you have greater period of fitness, or what we call health span, where you're still functioning individually, you can do the activities that you still enjoy, and then you remain healthy as possible until your death. And what you'll notice, whoops, I'm sorry about that. What you'll notice is the impact on lifespan is not that big. So we would call that a side effect of anything. But what we're really after is maintaining fitness and health longer in life. So what has been predicted as of the impact of geroscience is, this graph is showing, as we go out in time on the x-axis, the millions of people who are healthy beyond the age of 65. So this is where we are right now, the baseline. And if we delayed or cured heart disease well into the 90s or 100s, this would not shift that curve very much. The other, so that was cancer. For heart disease, still not much of an impact. But here is what would happen if we could actually treat the fundamental biology of aging. It would shift all of these diseases to later in life, or not so serious, or not life-impairing, and we would really have millions and millions of more healthy older adults. And this is estimated to improve healthcare costs by $38 trillion, which is astonishing. So here's two proofs of concept that the geroscience hypothesis could be likely to be successful. So many people age, as this woman is, housebound, checking her glucose on oxygen. Well, I decided I wanted to age like this young lady. She reached ripe old age while she's still out in the community doing good for people, and she has a really good wardrobe budget. So I think this would all be absolutely amazing. But the facts are that centenarians actually have 20 plus more years of disease-free survival. And on top of that, in the last 2 years of their life, they're spending a lot less on health care. So they're not in the emergency room, they're not in the hospital, they're not seeing specialists, they're not taking medicines. So human biology can do this. The question is, how do we get us all there without gene therapy? So another proof of concept of geroscience is the work done on metformin. So this is a drug, also called glucophage, used to treat pre-diabetics or diabetics. And we've been using it in humans for over 60 years. It's quite safe. And this is a meta-analysis, which was very clever to do. But basically, it's a survival curve after diagnosis of diabetes. You see that a diabetic on a drug other than metformin, they have a very steep slope compared in red to their matched non-diabetic controls. But what's interesting is diabetics who take metformin actually have a more shallow slope, so they survive longer than their matched controls without diabetes. And so it turns out, when we look back at the data, metformin is reducing the risk of diabetes or the complications of it, but it's also delaying cancer, it's delaying heart disease, and it's delaying cognitive decline in people on metformin. So this is, by definition, in my mind, a gerotherapeutic or a geroscience approach. One drug tackling 4 really life-threatening and quality of life reducing chronic diseases of old age. So this has led to the question, what in the world are we going to target to really hit the fundamental aging biology? And so this is really interesting because this came from a workshop organized by NIA that brought together a lot of experts in this field to think of what are the things that basically universally go wrong with aging. And this could be in nematodes or mice or rats or primates or humans. And there's just a lot of commonality. All of these things, DNA, telomeres, epigenetic changes, loss of proteostasis, very familiar to us. And they're all quite interconnected. So what's interesting is the same group redid this article a decade later, and we've made some advances. So there's nothing different in these hallmarks, but now they're put in a hierarchy. So the primary ones are really the fundamental molecular damage that happens in a cell. The antagonistic hallmarks of aging or pillars of aging are really the response to this primary damage. So all of a sudden your mitochondria aren't functioning, stress response and nutrient regulation is off, and cellular senescence occurs as a cell fate in response to all this damage. And then the integrative hallmarks are kind of third in the stream, and this is when things get really out of control, and I think intervention is going to be really tricky, because now you're talking about systemic problems like chronic inflammation, dysbiosis, and stem cell exhaustion, or cell-cell communication. So I come from a background of studying genomic instability, and very fortunate for me, this is a very potent driver of cellular senescence, and what we've learned over the last decade is that cellular senescence is really quite amenable to targeting as a potential geroscience approach. So we want to develop drugs that help clear senescent cells before you go from this antagonistic response to an integrative response. So for those of you who aren't familiar, senescent cells, this is a cell fate that is triggered in response to a whole variety of different types of stress, either internal or external to the cell. DNA damage is up there in that list. What happens is the cell triggers through signaling a fate where it turns on all these cell cycle inhibitors like P16, P21, P53, and this will prevent that cell from ever proliferating again. It's a stable replicative halt. And the point is, if you have a damaged cell, you don't want to make daughter cells because that would be tumorigenic. So this is a very, very potent tumor suppressor cell choice. There's almost always evidence of DNA damage in senescent cells, whether it's at the telomeres, the centromeres, or anywhere in between. And senescent cells also have a very peculiar morphology. Nuclear structure changes, the cells get big and flat. Many of the organelles are affected. But what's intriguing is they have this very potent secretory phenotype, the SASP, or senescence-associated secretory phenotype. So the SASP has all sorts of factors in it, incredibly heterogeneous, anything from chemokines to cytokines that are extremely pro-inflammatory, growth factors that confuse cell signaling, and lots of things that can disrupt the immediate extracellular matrix of the tissue. So you can think of these as a rotten apple. That's the best analogy I can think of. They're stressed, they're responding, and they're secreting things out into their environment that can really disrupt everything around it, like a bushel of apples. So what's important to me is, as somebody who has studied DNA damage, that to me is a cell-autonomous event. You have damage to your nuclear genome, the cell responds, and it apoptoses or it senesces. But I didn't understand how DNA damage could drive aging if only some cells are damaged. It's a very stochastic process. So what happens with DNA damage driving cellular senescence is you now have cell non-autonomous mechanisms of driving aging. They can secrete things into their environment, and they can go corrupt a neighboring cell that never even experienced DNA damage or other stressors. So this has helped us solidify the role of DNA damage in aging. What's really, really important for everyone to understand is senescent cell phenotypes are extremely heterogeneous, and we literally have, at this point in history, not a single specific marker. So you have to be very careful reading the literature. Nothing up on this slide is unique to senescent cells. And these cells are still at this stage very poorly characterized. So what we think is going on is senescent cells, they have this very pro-inflammatory SASP, secreting all these inflammatory peptides and proteins, and the goal of that is to actually ask your immune system to come in and clear those senescent cells, because they don't die on their own. They just hang out and they keep the environment inflamed. And so unfortunately, as we get older, our immune systems undergo immunosenescence or loss of function, and therefore are not as capable of removing these senescent cells. So what's also important about thinking about senescent cells as a target for therapeutics is there is definitive evidence that senescent cells drive aging. They don't just happen with aging, but they drive it. And this is very elegant work from the Mayo Clinic, where they used a transgenic mouse system where they took the P16 promoter, so one of those senescent cell cycle arrest genes, where messenger RNA goes up quite robustly with senescence, hooked it up to a suicide gene, which is caspase 8, and then a protein dimerization domain, FKBP. So what happens is you put this construct into mice. As they get older or they get diseased, P16 goes up, starts making this transgene, and then it's inactive until you add a drug to the mice. So you have to treat them with this AP20187 that will drive dimerization of caspase 8, and that is the activation that kills a cell by apoptosis. So in mice, you can get rid of senescent cells with this genetic tool. So when they first did the experiment, they did it in a mouse model of a progeroid syndrome, and you could see instantly that the mice that got the drug that cleared the senescent cells looked much healthier than the progeroid animals. Of course, they were asked to go back and repeat this in just normal mice. And so this is an experiment in aged wild-type Black6 mice. And what I like about this study, this is my favorite graph, is what you see in the animals treated with AP, which is the dotted line. You see a You see a 30% increase in median lifespan. Nothing happens to maximum lifespan. So that's squaring the curve. This is doing what we wanted to do. This is preventing all-cause mortality in mice until they reach a certain age. So a very important series of studies. This is work from Ned Sharpless that shows that senescent cells clearly accumulate with age. They were following the same mice over a period of time. And this is using a P16 luciferase reporter, so imaging the animals longitudinally in vivo. What you can see is there's incredible heterogeneity. And then when they get a tumor, it really lights up. But there's some animals that have a strong signal, and then a couple months later, it disappears. So this is really hard to capture. And this is just a graph showing total levels of P16 signal in the oldest group. So this is incredible heterogeneity. And what we think is these individuals up at the top would be at extreme high risk of multimorbidities of old age, while these down here look just as young as 4-month-old animals. So we need the tools to discriminate these groups of individuals. Another important point is you can pharmacologically target senescent cells. So this is a study we did with Mayo Clinic. And there's a couple of important points about the study. One is we started with very old animals, 20 months old. We treated them with the first cocktail of senolytics ever discovered. We only treated them for 4 months of age, for 4 months, and they only had to be treated biweekly. So that's another important point about pharmacologically targeting senescent cells. You don't have to have the drug on board all the time to have that impact. You would take it maybe once every 60 days, and then it takes 2 months for more senescent cells to develop. So this provides an incredible safety profile to this approach, I believe. And just 4 months later, again, you could pick out the animals that got the senolytics compared to their controls. And importantly, I think the study shows that the treated animals, I'm struggling with this, I'm sorry. The treated animals had maximal speed that was significantly increased, better hanging endurance, better grip strength, treadmill endurance, and spontaneous activity. So these are all things we care about very deeply. So I think there's a lot of promise for fighting frailty with the geroscience approach. So another study by Ned Sharpless shows that all of this is true in humans. So he figured out that the best way to measure cellular senescence is by measuring P16 messenger RNA in peripheral CD3 positive T cells. And so you can see as this is a cross-sectional study, but older individuals have greater levels of senescent cells. And this goes up logarithmically with every decade of life. Importantly, though, it's also modifiable. So here, current smokers have a steeper slope than former smokers and non-smokers. So doing things that increase your disease burden will increase your senescent cell burden. In contrast, exercise can lower your level of senescent cells, so we can head in the right direction with lifestyle interventions, let alone pharmacologic. So how did we contribute to this field? Well, it really started by me trying to understand the health impact of genome instability. So I want to understand what endogenous DNA damage that we produce in our cells every day of our life. In fact, approximately 40,000 lesions per cell per day that have to be repaired or cause consequences. And so studying this in mice led to this discovery of a human progeroid syndrome that's caused by mutations in XPF and ERCC1, which are two DNA repair genes. And what's very unique about this progeroid syndrome is it's virtually systemic. So every organ system is affected. What do these genes encode? Two proteins that bind and interact with each other in vivo, and they create a DNA repair endonuclease that's required for multiple DNA repair pathways. So what's important to note is when you knock out one, like ERCC1, you cannot find its binding partner. So really what all of the phenotypes in that patient and the mice that I'm about to show you is driven by lack of this holoenzyme, that it's a critical DNA repair enzyme. So I spent a decade of my life trying to figure out what's so special about this enzyme, and we linked it to three major DNA repair pathways. And so all sorts of DNA damage is accumulating, but we never do anything to the mice. This is all just normal metabolism in healthy animals. And then the other thing I wanted to point out to you is these two gene products are only ever found in the nucleus and never in the mitochondria. So everything we're seeing is a direct consequence of DNA damage and not in the nucleus and not protecting it. So this is the mouse model that I made of this disease. It's been very serendipitous. These animals reach adulthood looking pretty healthy, but they decline very, very rapidly. And they express about 5% of the normal complement of this holoenzyme. So these mice, without doing anything to them, within their 6 to 7 month lifespan, develop all of these different age-related diseases. It's pretty dramatic, keeps us really busy because I'm an expert in none of them. So we love collaborating and happy to share mice or tissues at any time. So this is all spontaneous age-related diseases related to failure to repair DNA damage. And to prove that, we elicited the help of Yinsheng Wang, who builds these beautiful LC-MS-based assays to detect endogenous oxidative DNA lesions. So here we're looking at 4 cyclopurine adducts, which ERCC1 XPF is required to repair. And when we look in renal tissue, each of these bars is a sum total of these 4 adducts at different ages and genotypes. And you see at a young age that the mutant animals really don't have increased levels of these particular oxidative lesions. Now, these are just surrogates, and there's obviously many more lesions to measure. But by the time the animals are frankly progeroid, they do have a significant increase in oxidative DNA damage. But what's really intriguing to me is this bar over here. These are wild-type mice that have a full complement of DNA repair. And over their lifetime, they accumulate the same amount of this oxidative DNA damage. So this is a physiologic way of getting to aging. It's just happening faster when you take away DNA repair. So what we did is we tore apart the mice and we looked at senescence markers in a variety of different tissues. And we did this for P16 messenger RNA as well as other markers of cellular senescence that are well accepted. And what's really fascinating is here's what old wild type looks like. So the aorta has more senescent markers than inguinal fat, liver, et cetera. But the rank order is exactly the same in the ERCC1 deficient mice. So we really think this is, again, a compression of what happens with normal physiology. It just happens very quickly. So we went on to then say, okay, if you can genetically deplete senescent cells, can we do this pharmacologically? And this was a study done with the Mayo Clinic where we just started out with transcriptomics and looked for genes that were differentially expressed in senescent cells. And then we would pick our favorite targets and do siRNA to knock them down to make sure that they were killing off senescent cells, which are in black, as opposed to proliferating or healthy cells, which are in white. And what's been very intriguing is that if you use bioinformatics to sort of collect these clusters of genes that are differentially expressed, you find many things that are very, very familiar to oncologists. So cell cycle regulators, serpins, PI3 kinases, et cetera. So the first senolytics that were ever described were dasatinib, which is known to target ephrin receptors, SRC, BCR-ABL, et cetera. Quercetin, which inhibits this PI3 kinase and other kinases and serpines, and then direct BCL2 inhibitors. And so what's intriguing here is that we can repurpose cancer drugs as senolytics. And on top of that, we do the best job when we get sloppy drugs. So those that don't have a single molecular target, but actually hit a number of nodes that help keep senescent cells alive, like tumor cells. So we have, at iBAM, created a number of screening tools. We take mouse embryonic fibroblasts from our mutant animals. We just expose them to atmospheric oxygen, so again, very physiologically relevant. We get cultures that are about 50% proliferating, 50% senescent. We put on drug libraries, and then we do kind of a medium throughput measurement of senescence-associated beta-galactosidase, which can be done with a fluorophore that's a substrate for beta-galactosidase. So we measure the number of total viable cells and senescent cells, a lot of validation after that. And this is kind of how the profile looks when we're done with the assay. So what we've plotted here is the relative amount of senescent cells after drug treatment compared to total cell number. So this group of drugs had no impact on senescence whatsoever. We ignore them. This group reduced the amount of senescence, but not the amount of cell numbers. So we call these xenomorphics. They're somehow altering the senescence phenotype, but not directly killing these cells. And rapamycin is right here among these, and we know that's exactly how it functions. It's not going to kill a senescent cell, but it makes it less inflammatory. And then these are the winners down here that we called senolytics as a new group of drugs. They knock down the number of senescent cells by killing them. So there's reduced cell number and senescent cell number. And amongst these were HSP90 inhibitors. So again, something we can repurpose from an oncology clinic. And this has been reproduced by a number of laboratories. So where's the field now? We have a ton of senotherapeutics that are out there. They're really interesting as classes of drugs. Some of them are natural products, which really does not surprise me at all. Mother Nature has always done a great job of making potent carcinogens as well as cures. And a lot of these are flavonoids, which are incredibly unattractive to big pharma because they're very sloppy drugs, just like dasatinib. Some of these are cancer chemotherapeutic agents that have been repurposed. Some are frankly senolytic, and others are much more xenomorphic. We've now got folks in the field working on much higher throughput screens, vaccines, CAR T cells, and PROTACs. So there's lots of activity in this space. We do know at this point in time through the work of dozens of laboratories that if you use either the genetic or pharmacologic approach to kill off senescent cells, they work in a variety of mouse models of human disease. And I think this is really exciting because it really says this is geroscience, right? It's not just curing a liver disease or a brain disease, but it's targeting multiple organs and multiple age-related diseases. The Alzheimer's study is really exciting because we definitely need new therapies for Alzheimer's disease. And I'm going to tell you a quick story about COVID-19. So we've gone from discovery of senolytics. In less than 10 years, we now have 40 clinical trials ongoing in this space. So a quick story about COVID-19. Early in the pandemic, we made this funny discovery, and clearly this started because when we looked at all the disease that puts you at bad outcome risk in getting SARS-CoV-2, then all of them had in common senescent cells. So we asked, could senolytics work in this case? Well, we have a very unique system at the University of Minnesota where we take our pathogen-free experimental mice and we expose them to pet store mice. And we call this normal microbial experience because we do not live in a sterile environment at all. We're constantly exposed to environmental microbes that don't kill us. So when we did this experiment with young mice, 90% of them survive. And so this is great for immunologists who want to study a more humanized immune system. But when we take the aged wild-type mice, 100% of them died within 2 weeks. So it tells you a lot about kind of the conditions of how we're working with our mice. And I think there's room for improvement or just alternatives here. We figured out very quickly that the thing that was killing the old mice was actually a beta coronavirus. So in this case, it's MHV or a hepatitis virus. But we could see in the animals just before they died, that was the only of the pet store microbes to which the animals had all raised antibodies. We found pathognomonic lesions associated with MHV in the liver of old mice, but not young, because they could clear it. And then our immunologist friends did the most elegant experiment where they gave our young and old mice a sublethal dose of MHV and then exposed them to the pet store mice quite a bit later. And you see that rescued 100% of the animals, which is the blue bar. So MHV is clearly killing them, and that was a beta coronavirus. So then we tested our senolytics, and in this case we used fisetin because we were thinking about old people. This is actually something that comes from strawberries. It's a natural product that's available over the counter. So very attractive as opposed to a cancer chemotherapeutic agent. And so we tried to do this in a translational way where we exposed the animals to these pathogens they'd never seen before, and then we treated intermittently 3 times, 3 days in a row. And this rescued 50% of the mice. We don't know why it wasn't more, but we did know drug optimization in terms of dosage and things like that. So this has led to now clinical trials in sepsis in older adults and COVID-19 patients and nursing home patients. We think the reason that senescent cells contribute to bad outcomes is because they cause cells to overreact to infection. So this is in vitro data where we've taken senescent cells in yellow or non-senescent cells in black. And if you challenge them with a bacterial LPS, then you basically get this huge overshoot of production of IL-1 beta. This also happens when you look in animals that are challenged with LPS. So they think they have an infection, and IL-6 goes through the roof compared to young animals. This also happened in the NME paradigm. Again, IL-6 in old mice was much increased compared to young animals. And if we treated the animals with fisetin, we could bring that curve back down. So we really think senescent cells plus the immune response to a virus are creating the cytokine storm that really kills older individuals. And so I think of this as senescent cells just confusing the heck out of your immune system. And this, I think, is some of the strongest proof for geroscience. I can imagine chronic diabetes, of course there's senescent cells that are going to build up, but you have senescent cells and you have a completely new stressor and you can't respond properly. So it argues that interaction or intervention before a challenge could potentially really improve overall health in older individuals. So the other thing that we've done is we've done tissue-specific deletion of ERCC1, trying to ask the very naive question of which cells are most deleterious when they become senescent or damaged. And much to my delight, as we've done these experiments, every single situation we have come up with a disease of old age. So after 20 years of fighting with various groups, I think there is really strong evidence that endogenous DNA damage does contribute to age-related diseases of virtually every organ system. So a quick story about the hematopoietic system, because this is the dangerous one. So we deleted ERCC1 using VAV-I-Cre which targets every cell in your immune system. And what we saw is a progressive decline in peripheral blood counts. And so white cells, they get a leukopenia, but not until adulthood, and then it gets progressive from there. At a young age, we do not see markers of cellular senescence in immune cells from these animals, but by the time they're older, using the Ned-Sharpless assay, we see very high levels of all of these different markers of cellular senescence. And this is quite comparable to 2.5-year-old wild-type mice, which are shown in yellow compared to the mutant animals in red. When we did CyTOF to try and confirm this, we see that all different types of immune cells, whether they're T cells, B cells, NK cells, or myeloid cells, they all show signs of cellular senescence. So we've really tried to hone down on which is the key driver of all of this. But the surprise in all of this was actually that when we looked across solid organs in these VAV-ICRE mutant animals, P16 was actually elevated in virtually all of them. So here we have only DNA damage in immune cells, only senescence in immune cells, but driving secondary senescence virtually everywhere they go. And we could show that, in fact, using FISH, so trying to detect P16 messenger RNA, it's actually in the parenchymal cells and not just tissue-resident immune cells. So this is localizing with KSP cadherin, which is a marker of renal tubule epithelium. Remarkably also, this senescence, secondary senescence, was enough to drive disease in many solid organs. So the VAV-ICRE mutant mice in red here have elevated liver function enzymes in their serum, as do old wild-type mice. They have proteinuria, just like old mice. They have serum amylase that's elevated. They have disc degeneration, and they do not respond well to a toxin causing muscle damage and regeneration. And in fact, these are the VAV-ICRE mice in red. They actually have a shorter lifespan. So Ned-Sharpless has taught us that there are senescent immune cells in all of us as we get older, and this is telling us they're not doing us any favors. Just to prove this point, though, what we did was we took splenocytes from our mutant animals or aged wild-type mice, and we transplanted them into a P16 reporter mouse that was young and healthy, measured the IVUS signal using Ned's mice, and then what we find is post-transplantation, the signal in these recipient animals goes way up. So for 5 weeks, actually, they had increased senescence just by transplanting senescent immune cells into them. And they also had elevated levels of P21 in all of their solid organs and circulating levels of, that were very high of inflammatory cytokines and chemokines. So immune cells are definitely a mediator of secondary senescence. So a quick summary here. I hope I've convinced you senescent cells accumulate with age, promote age-related diseases, drive aging, and can be therapeutically targeted. Senolytics, pharmacologic or genetic approaches are really reducing adverse outcomes in old organisms upon infection with a novel pathogen. I think these studies support the geroscience hypothesis and the strength of this approach. And I think immune cells are really dangerous. And so we're thinking about all the different applications we could possibly apply this to. So we're studying senescent immune cells in infectious disease, cancer, transplant medicine, HIV, you name it. So with that, I'd just like to forecast in the future just for a couple of minutes here. Geroscience, I think, has a lot of room to grow, and I'm really hopeful that a lot of people take this on, regardless of whether you're in an institute that's not focused entirely on aging. I think this is relevant in a lot of arenas. So this is a beautiful paper put together by George Kuchel and Nir Barzilai, where they looked through a ton of literature and found FDA-approved drugs, which are listed here on the left. And there's already evidence that these drugs, no matter how they're used, which is very diverse applications, actually can affect multiple organ systems. And there's even data that they affect the health span or lifespan of human beings. And there's also a ton of preclinical data that supports all of this. And there's also evidence that each of these drugs target multiple hallmarks of aging, which is fantastic. So we have things out there that we need to deploy, and we just have to figure out the right design of a clinical study. There's a lot of attention in the longevity landscape. There's a number of companies that have popped up. There's a number of granting organizations, social catalysts, mega foundations, and industry vehicles. So I'm happy to share these slides with anybody who wants them if you need a list of ways to get funding. So another thing I wanted to point out that's very interesting is ongoing geroscience clinical trials right now, they're all targeting these antagonistic pillars of aging. So none are up here and none are down here, and I think this is probably the way to go. So my view is that these primary hallmarks really require specialized laboratories to measure them correctly. It's not trivial to measure telomeres and epigenetic changes, DNA damage, and autophagy. I suspect all of these are so critical to life that they've actually been evolutionarily optimized. And so targeting them pharmacologically is going to be really tricky, because if you overshoot, you're going to have bad side effects, is my prediction. There's already clinical trials in Alzheimer's, and I just wanted to point out this is a tour de force, because this is with interventions with dasatinib and quercetin, those first senolytics, published in Nature Medicine last year with just reporting on 5 patients. So even baby steps are attracting huge progress. So this is my lab. I'm very proud of all of them. I've got great postdocs who are carrying all of these projects, a whole mouse team, a neuroscience team, bioinformaticians, a clinical researcher, 3 graduate students, and a bunch of undergrads that I couldn't live without. They do all the work. I wanted to say a quick message about Judy Campisi, who recently passed away. She is actually one of the founding leaders in the study of cellular senescence. She was at the Buck Institute, and my dream was that she was going to get a Nobel Prize for this, but unfortunately that won't happen now. So we're very, very grateful to all of our funding sources, including, most importantly, many agencies at NIH. And I wanted to say just one word. This is my last slide. The SENNet Consortium is a director's common fund initiative bringing together talent from every aspect of science to study, define, and figure out how to target senescent cells. And I really, really love this big science. So if you're interested, let me know, because we'd love to have you as part of SENNet. So thank you very much for your attention, and I'd be pleased to take questions.
Thank you, and please, anyone in here, please use the mics if you have some questions. There are a few online. Wanted to repeat the CME code 50113. 50113. So a couple of questions are kind of connected to each other. I'll see if I can combine them. Very interesting talk. If aging is highly correlated with increase in senescent cells, how many senolytics are flavonoids found in various foods? And why does diet not affect risk of various diseases you mentioned as much as age? And similarly, is there any overlap between the senolytics and the, say, so-called Mediterranean diet?
Great question. So yes, there actually is great literature out there showing that flavonoids and foods that contain a lot of flavonoids actually affect multiple diseases of old age. So the Japanese tend to eat more flavonoids, and it reduces heart disease, LDL levels. There's some link to cognitive impairment. So I think a healthy diet with lots of fruits and vegetables that you can get these things is much safer at this point than the fact that you can buy some of these over the counter. Because let me remind everyone that these over-the-counter supplements are not federally regulated. We have no idea what's in them, and they can lie all they want on the label about purity. So the Mediterranean diet and fruits and vegetables, definitely the way to go.
Okay, that might fit into this next question, actually, another pair. One of the side effects of BCL2 and TT53-MDM2 interaction inhibitors is neutropenia. How such side effects can be realistically managed? How can such side effects be realistically managed if such drugs are used for trying to target senescent cells?
That is a very good question. So there's big problems with BCL2 inhibitors. They cause not only neutropenia, but loss of platelets. So there's bleeding disorders associated with them. That led the company Unity Biotech to target senolytics locally. So they use them only for knee joints or for macular degeneration. And so if you can contain where that drug goes, that's one approach. The other approach that people are using is to develop like PROTACs, or ways that you can target very specific types of cells with a BCL2 inhibitor. P53 inhibition, in my mind, is not a great idea at all. I mean, that is the guardian of the genome. And so we have actually put our mice into a P53 deficient background, thinking we'd get them to explode with cancer. They didn't. They just aged differently. And overexpression of P53 has clearly been shown to drive aging as well. So those, I don't prefer to target specific molecules like that. I kind of like the approach of these sloppy drugs that might be doing a little bit of antioxidant work, signaling work, as well as affecting senescence phenotypes.
Okay. Part two of another question. You mentioned that there are 40 or so in clinical trials. At what stage and how long do these studies on aging, clinical studies on aging, last?
This is a really good question, and this is where we need the collective wisdom of the entire field, and everybody in NIH is designing the proper trial. So right now, the FDA does not recognize health span or lifespan or aging as any sort of useful endpoint in a clinical study. You can imagine the size of the study it would have to be, the duration, the expense. It's just not realistic. So all of these trials are aimed at very specific things like frailty, Alzheimer's, cardiovascular disease, and they're designed to be shorter. And the first goals are to prove that there's safety, using an adaptive sort of protocol so that you can figure out the right dose, and target engagement is very important. And then basically the secondary endpoints for all of these are then health-related and metabolically related. But we have to prove these drugs are safe and have some effect. The one trial that's been negotiated with the FDA is the TAME trial, if you've heard about this. So it's with metformin. It will test metformin in people with a single disease of old age and measure the time to the second onset of disease or death. And that usually happens in 2 to 5 years. So that will be a short trial. If that trial is successful and proves that metformin in a double-blinded, placebo-controlled manner staves off comorbidities of old age, then FDA will open the floodgates and we can do real studies.
Okay. Your clever pet shop experiment made me think of this one. Is there any connection between pre-probiotics and changes in the senescent cells with age?
There's definitely a connection, and that is not my area of expertise, so I don't have more specifics to that, but incredibly understudied area for sure. And I really do think we need to think about old mice, their microbiomes, their exposomes, everything, to get a more humanized system to really test gerotherapeutics.
Okay, and I figured this was coming. Fasting, can you comment on how different types of fasting influence senescent cells?
Yes. So it's very clear that caloric restriction and exercise are the two things that work right now and are safe. Caloric restriction, there was this very landmark work in non-human primates done at the intramural NIA program in Madison. And they got very different results in their studies. And it turned out it was actually not caloric restriction necessarily, but how long the animals were exposed to food during the day. So caloric restriction is uncomfortable. It's just, it's not going to be widely adopted. So what we're talking about right now is either intermittent fasting, so eat every other day, or compressed eating. So pick a window of time in your day when you can eat all of your calories. Say 1 o'clock lunch, 6 o'clock dinner, done. And if you can do that, that has health benefits, and that's been shown up through primates, which is as good as you're going to get right now. So,
okay, let's finish with one question from the audience, please.
Hi. I was interested in one of your first trials, first slides, where you said, without gene therapy. Now, gene sequencing has advanced to the point where sequences can essentially be done overnight.
Yep.
And on the other hand, you said 2 to 5 years is a short trial. That just doesn't add up. I would like to suggest instead that you get a whole gene sequence, you get it analyzed. There is a huge literature of sequences to compare that sequence to, including in Alzheimer's. You fix, I'm oversimplifying, but you fix the broken genes, or you turn them off, or you replace them with functional genes, and you do those manipulations, and then you put the manipulated cells back into the person. And all that can be done literally in the matter of a week or so. So, do you have a problem with that sort of approach?
No, I think you raise an absolutely excellent point. I was actually making a little bit of a quip because there's a company out there that wants to make that P16 caspase 8 construct and put it into humans, and I'm not going there. I don't want to try that. But we do have a very generous grant from NIA that's to look at the genes of centenarians and find the variants that correlate with healthy aging, and then try and mimic that in an animal model, as well as come up with drugs to target that pathway. Gene therapy has a long way to go still. I mean, because we don't know which organ ages the fastest, and so how do you target a specific organ with that gene therapy, or do you really want it systemically? How long do you want it to express? So I'm not convinced that's a faster way, but certainly genomic analysis and transcriptomic and all this omics is going to give us a lot of information, and that's what SENNet is all about. So
it will also provide work and important jobs for
Absolutely.
young people. So I would not worry about young people being able to find work. And I'll leave it at that, but I'll be glad to talk with you later.
Sounds great. I agree. We couldn't do all this bioinformatics without young people. Thank you.
Well, thank you for coming, everyone, and thank you, Laura. Great talk. And if you join us outside the auditorium for a little reception, we can talk more with Dr. Niedernhofer.